So Natalia did a wonderful job of describing that I'm from IMET, which for those of you who have been around for a long time, you might recognize it as COMB, which is right here in Baltimore's Inner Harbor. If you ever come visit Baltimore, let me know. I'd be happy to show you around. We're about 2 piers down from the aquarium. And these two wonderful people with me in the photo, Jennifer and Tori, contributed a lot to this research. Our lab is larger now, but we have never gotten together for another photo. I keep trying. So throughout this talk, there are actually a lot of current collaborators and co-authors that play a role. Just to put this out there first, but a lot of the work couldn't be done without my collaborators, Carolyn Friedman and Kim Reese at VIMS, and also current and former members of my lab. So as Natalia alluded to, I work on marine host-pathogen interactions. So maybe not in this crowd, but I often say that I'm the only person that goes to the beach and naturally looks for sick organisms. So here, I'm looking for 6 sea stars, so I've worked across a wide range of marine host pathogen interactions. And classically, this has been represented by a triangle or a Venn diagram. I really like this triangle because the triangle represents change. And I often think of it as a lens of what are the changes to the host or the pathogen. So is there a change in host density? Is there a change in pathogen virulence that lead to disease? And are there environmental drivers involved? And I'm not saying this strictly in the climate change per se, but there are often environmental correlates of marine disease, particularly in marine invertebrates, where a lot of what I study. So I have worked across a wide range of marine host pathogen interactions, starting with, for my PhD, and also work now in the lab with Pacific oysters and the oyster herpesvirus. We also work a lot on Zostra marina and this Labyrinthula zostra, which is the causative agent of wasting disease. And we also look at interactions between the oyster and eelgrass. And we still do a little bit of work on Gorgonia ventolina and a couple of different pathogens, both in a Planicitarian pathogen, which is similar to this Labyrinthula, which is how I got involved, and also Aspergillus sodowii. And we've also had some projects focus on physiology of corals across different gradients. But if you have questions about those projects, please ask me later. I will focus on our work on the Pacific oyster and the oyster herpesvirus today. So the Pacific oyster is an interesting species because it is actually native to Asia, but has been moved all around the world. I often talk about this on the West Coast. People on the West Coast don't realize this is a non-native species that is being cultured there. And it is the most popular oyster species globally for oyster aquaculture, worth billions of dollars each year. And it's a fast-growing and hardy oyster, often replacing native oyster species production. And in some areas, they're naturalized, so they're able to reproduce. In other areas, depend on oyster hatcheries. So just to orient you, because different life stages will come up as I give my talk, typically we have our adult oysters, which are broadcast spawners, at least for some oyster species are actually brooding, but not the Pacifics. They'll release their egg and sperm in the water column, and they'll develop, and they'll have this larval form for approximately 2 weeks. And this is really how the oyster colonizes new areas. Then they'll settle to the bottom and become spat. I may use the word seed interchangeably for spat. That's kind of an industry term. And honestly, they can be reproductive and up in earlier than this, 2 to 3 years, which is printed on the slide. So in culture settings, you can actually, in case you're not familiar, because they're broadcast spawners, you can strip spawn them, and you can make all sorts of different types of lines of oysters. In fact, this has been a very popular solution to disease issues, for example, in the Chesapeake Bay and other places. And for the industry, they use lines or stalks, and a lot of the growers know which ones do well. And for the interest here, some of the work I'll present is in small seeds, some in nursery-size animals, and then in the field, we often work with off-bottom culture. So the primary oyster species grown in the US, and the primary reason I bring this up is I'll actually talk a little bit about most of these species in the talk. Of course, I've already introduced Pacific. Most of you are probably used to the eastern oyster, which is on the eastern seaboard in the Gulf of Mexico. The native oyster on the west coast is the Olympia. It's a much smaller oyster, even though this picture makes it look larger. And then we also, on the west coast, Kumamoto, which are kind of a cousin to Pacifics, are another species. They're the little, deep, yummy ones. And then the European flats, which the name suggests came over from Europe. So what is this virus? So this ostrich herpesvirus 1, the reference strain was characterized, so it was sequenced from infected oyster larvae in the early 1990s. And morphologically, it's similar to herpes viruses. So this is OSHV1, this is herpes simplex virus 1. And the genome is about 207 kilobase pairs. And it's grossly similar to the other animal herpes viruses, and it has unique regions, unique long and short, flanked by terminal and inverted repeats. In OSHV1, there's this extra region X. And multiple strains of this virus exist, both that specifically infect Pacific oysters as well as other species, and there's actually a lot of genome variation that will come up later in my talk. And it's actually, when you have a very large amount of this virus that we work with, it's pretty easy to sequence. And you can also pretty easily do some gene expression work with it, but I won't focus on that today, but I'm throwing that out there for Maya. So you've all been wondering if you can safely eat oysters now. And the answer is yes, and the answer is no, this virus does not infect humans. And hopefully you're looking at this tree and this makes sense because OSHV1 branched off very early from the terrestrial vertebrate viruses like the alpha, beta, and gamma herpes viruses that include the human herpes viruses, and those that infect fish and amphibians. And in fact, it really has only one gene that's similar across all of these now. Well, I'll get to that. There's one more virus recently that has been similar, the abalone herpes virus, which I've added to this tree. But it was recently reclassified as the order Herpes virales, with Tomales in the family Malacca-herpes viridae. I think probably because of these large differences in the genome. So why do we care about this? So OSHV1 causes mass mortalities of Pacific oysters. And this, in the time of this reference strain that I talked about, it was generally in larval and seed oysters and in juvenile oysters. Its host range is not restricted to Pacific oysters, and in fact, we don't fully understand the vectors and reservoirs of this virus. It progresses rapidly and can kill up to 100%, dependent on the host genetics, et cetera, and some of the other environmental parameters. And once established in an area, generally mortalities happen every year. So every year in areas, there are seasonal mass mortalities. And it can be transmitted within and between life stages, both horizontally and vertically. And this virus is culturable, so there's not molluscan cell line. So later I'll discuss how we're able to really work with this virus in the lab, but for a long time that caused severe difficulties. So what does this look like in the environment? I'm just showing you an example from my own work, but this is kind of a depiction of what happens in a lot of areas where there is OSHV1. So on the x-axis are 2-week intervals between surveys of our oyster mortality. On the left y-axis is percent mortality, which are these hatched bars. And what we found in this particular year, and degrees Celsius are on the right y-axis, is following the high mean temperatures of the year, which I realize is nothing since we're in Florida, and the maximum temperatures, we first detected this virus. So no virus had been detected before mid-July. We'd been doing, at that point, conventional PCR the whole summer, followed by these mortalities. And these mortalities, this is a mean bar of the instantaneous mortality of the year. And the farm I work with would describe that they lose 50% of their oysters every year. So this is highly problematic. But again, there's this sort of seasonal environmental pattern that's actually very common with marine diseases. So how do we diagnose this disease? Well, classically, it was with electron microscopy, which I'm sure many of you might realize this is a bit like a needle in a haystack. So this is a beautiful image from one of my transmission studies from my PhD, where we were finally able to see these beautiful herpes viruses lined up between two cells. But more commonly now we use qPCR, and this is just an example of a typical qPCR standard curve where we're going from 3 to 3 million copies. And this is probably a very high efficiency and a very high R squared as well. But so a low copy number sample might be down here, which makes sense. 3 copies per however much tissue you put in, where a high one might be up in here or even higher. So early in time, in the early 1990s, there have been descriptions with EM in France and New Zealand, followed by the sequencing of OSHV1 in France in 1993, and had been associated with sporadic mortalities of larvae and seed. We detected the virus for the first time in Tomales Bay in 2002 and have detected it as early as 1995, followed by detection across multiple Asian countries, but with unknown mortalities. So something changed in 2007 and 2008. There were severe economic devastating mortalities of all life stages of Pacific oysters in France by a variant they named OSHV1 microvar. What is an OSHV1 microvar? Well, it's actually fairly confusing the way it's been described, but it's based on specific genome regions. And importantly, it can kill oysters of all life stages. And we now understand it's not just this one microvar, but probably many microvariants. And I'll get to this, you'll understand better on the next slide. And again, I've already said this, but it's all based back on that reference genome that was first sequenced. And it is also the causative agent of Pacific oyster mortality syndrome. If anyone has heard of this, particularly in Australia, causing mass mortalities of Pacific oysters, this is one and the same thing. They do not call it herpes in the media for obvious reasons. They call it POMS. And there's high level of concern about the spread of the OSHV1 microvars. And why is that? So we come back to this map. I've already mentioned it appeared in France in the late 2000s. Well, it then was spread throughout Europe. So with a lot of it with oyster transfers likely, and into Northern Europe, probably from that larval stage of oysters that they've been getting feral oysters into Northern Europe. But then in Australia and New Zealand in 2010, a different variant than the original microvar was detected in New Zealand and Australia, causing mass mortalities in their oyster aquaculture, followed by movement of about 850 miles to Tasmania in 2016 and actually to South Australia, I believe, in 2018. So no one knows how these viruses came to Australia and New Zealand and why they emerged when they did. We do know that now we have a better understanding there are some of these other variants in Asia, but there's not a lot of information coming out of Asia about potential mortalities. So this sets the stage for a very interesting emerging virus situation. And in fact, it has been a high level of concern. And then very recently at the end of my talk, I'll talk about a recent detection in San Diego, which I have classified as a microvariant. So I often get asked about management, regardless of whether I'm giving a basic science talk or not. And one thing to know is generally with marine disease, but also OSHV1, it's really not practical to cull. And we can't vaccinate oysters, so they don't have an adaptive immune system. So it's not like we can give them a flu shot like we get. But there's been some really interesting recent work with a viral mimic, poly-IC, if anyone has ever heard of it, that does enhance survival, even giving it to the adult oysters before they spawn them. Can restrict the movement of animals, and also educate end users on risks if OSHV1 is detected and improve the speed. So if I'm talking to oyster farmers, I will tell them, please send in your samples to the diagnostic lab if you see an issue. We can breed for resistance and also look at genes involved in OSHV1 resistance. We can determine species more resistant to OSHV1, which I'll talk a lot about today. And finally, water treatment can be used in closed systems, but I think most of us realize that oysters really do much better in the marine environment than they do in closed recirculating systems. So breeding for resistance has been a good practical measure. So breeding programs exist in France, New Zealand, and Australia, and likely other places. The resistance to the reference strain in France actually confers resistance to at least some of the microvars. And these resistant oysters can actually limit infection and eliminate the microvirus from their tissues. And my collaborator in France has pointed out groups of oysters to me and told me those were the really good ones that they don't get infected at all, so, which is quite interesting. And throughout kind of everywhere where this has been a problem, resistance is proportional to size. So smaller oysters are more susceptible. Coming from our work in California to work in Ireland, France, Australia, pretty much all over the world. So when I talk about these mortality events, this is an example. We can use non-selected lines. So one of the caveats here and is that when you don't have a disease, it's really hard to use selective breeding. You could select specific lines, and this is just an example. These were actually really small oysters. These are smaller than we usually like to plan out, but that's what we had that particular year. So line A ended up around the 40% mortality mark, where line B had much higher mortality. So this is kind of an example of what we often see. And at the same time, we saw, this is the copies per oyster, because the oysters were so tiny, they were like the size of my fingernail, that we could extract the whole body from 0 to 1 billion copies per oyster. And in the midst of the mortality period, you see that the copy numbers are extremely high. Some of these oysters had between 10 and 100 million copies in their little body. So you can understand how transmission could be so risky and how this could happen. And this is considered the less virulent strain. So I may not have made this point clear. So my work in California is considered a less virulent strain than the microvirus. So our objectives of about the last 3 years have been looking at both field trials to identify oyster lines and species with differential susceptibility, and also laboratory trials to examine differential survivor, and with the thought that there'll be species and family lines that'll demonstrate differential survival in the face of OSHV1 and OSHV1 microvirus exposures. So to start off with, I'll just briefly describe some of our work in the field in California, where we're all happily here early in the morning. So we work in Tomales Bay, California, which is about an hour and a half north of San Francisco. I think this map shows it pretty nicely, and we work on a couple of sites. They're actually pretty similar to the sites that I described in an earlier slide, and here also is here. And we also had an oyster holding area outside of the OSHV1 area, so we actually planted out all of our families 3 times. So, and that was primarily because we know that there's often, I hate to say a sweet spot, but there's a time where oysters are particularly susceptible when they're outplanted, when the virus is out and causing infection. So we used 4 different stocks of Crassostrogygos. I won't get too specific into details, but I can describe better if anyone is interested on what those are. Crassostrogygos, and then this native oyster species. And these are just, I think this was the midpoint of the summer. I think we ended up with even more real estate on the farm. I felt like for a while that I had my own oyster farm. So this is us out in the field doing our monitoring of the species and stocks in Tomales Bay. And Melina is very nicely showing us how she has dumped all of the little oysters out of the bag onto her tray. Then she's counting, so she's counting into piles of 10, and she would give me a number of dead oysters and also pull out all of the dead oysters. And for some reason, these guys are always particularly happy early in the morning. I'm not sure why. And then they would pass along 10 of these oysters. I would take a photo of them for size, and then I would flash freeze them so we could do both our qPCR and any future potential other RNA work. And finally, these guys are happily in the lab doing all of their tissue sampling for qPCR. And I forgot to mention, in this particular year, we did a sentinel. So we went out every week, which is shown here on the x-axis. The weekly intervals are not perfect because of the tides, but we used both of those sites. In the next couple of slides, site A is in blue and site B is in gray. And we used one particular stock, and we monitored over the summer. And as we were monitoring, we could, we could see some increases of copy number, but it wasn't really until about week 8, so around July 20th. And I had some colleagues coming in and I said, oh, this is perfect time, Carolyn and Kim, we're going to see an outbreak. And in fact, we did. They came out, I think, on the 25th of July, and very happily we did. And clearly we can see this increase over the summer. Unfortunately, right when it was at the height, we could not get to the second site on the same day. But clearly this group had very, very high copy number at this time. So as I mentioned, we planted each of our stocks or species 3 times, 3 different plants to capture what we knew would be some differential mortality, both between the stocks and also the outplant sites and the timing. So this is just an example of one of the groups, Midori. And at site A, it had pretty consistently low mortality. At site B, it had a big spike right after the second outplant. So just to orient you, when I show the next part, there'll be all of the groups like this. Again, here they are on Murderer's Row, counting all the oysters. It was an exciting day out there. But then we came to kind of the final cumulative data, and again, the second outplant had particularly high mortality, actually at both of the sites. And from here, I highlight these 4 groups because they will be in our lab experiments as well. So brothers and sisters of these groups we planted in the field, we also took to the lab and did experiments. So 3 of the C. gigis groups, 1 Sycamia group. Further making sure you know this, calling it out screamingly. And as I previously mentioned, this is considered a less virulent form of the virus. This was 500 oysters 3 weeks before. This particular bag, number 88, lost 99% of this particular stock. And poor Blythe ended up counting them every single time. So even though this is a less virulent form of the virus, it does indeed cause mass mortalities given the right timing. So why am I showing you a picture of Tucson, Arizona? I actually just came from Tucson, Arizona. And the reason is we've been conducting our experiments in Arizona purposefully in a biosecure location in actually a shrimp pathology lab, 200 miles from the ocean, purposely in strict quarantine. The last thing that one of us wants to happen is have one of these viruses get out. And it's in part because, we brought in virus from both France and Australia. So working with the USDA, APHIS, as well as the state veterinarian of Arizona, we brought in these viruses as a proactive approach to prevent and to understand potential species and stocks that can be grown. So here we are in Arizona, and one of the first things we have to do, so we received little tubes of virus. One came actually on ice and the other came frozen. And we use susceptible animals. Actually, these are that line that had 99% mortality, and to make larger stocks of virus. And I call them homogenous because they're somewhat purified. They're homogenous of oyster tissue that have been filtered through a 0.2 micron filter. And this was done by sampling moribund animals, so animals that really were dying so they wouldn't be too gross. And the challenge types I'll describe, there's 3 different challenge types, so bath, injection, and some natural water exposures, which have, there's good reasons to use each of them. And if you look really closely here, these 2 guys, if I tapped on their tank and they didn't close up, they were probably just in the process of dying. So that's actually what one of the dying oysters would look like. And we were really dependent on qPCR to give us a viral load and to understand what we were injecting or exposing animals to. So we did a first comparison in a bath challenge with 9mm naive Pacific oysters. And no controls died. They're not on the graph, but you're probably already noticing time and days here and survivorship here that very rapidly the oysters died. So very rapid decline over the 6 days. And though there was a statistical difference in the mortality, I don't think any of us would be very happy if we were farming these oysters and saw such large mortalities. And the qPCR data backed up this challenge result, so we had very high copy numbers expected in these animals. So we followed this up. So these are the groups that I described earlier, the CG1, 2, 3, the Crassostrea, and just in case anyone is familiar with oysters and is wondering what life stage I've included, kind of the general size, it's also somewhere about here. And based on the field data, I had some hypotheses. So I thought this one would have low survivorship, so that matched with the bag 88. This one high, that was the first group I showed you. And then this one moderate, and finally the Crassostrea, which I forgot to point out, had very high survivorship in Tomales Bay. And then we actually used 2 lines that were produced at the Virginia Institute of Marine Science, Debbie and Lola line, which you may be familiar with if you're familiar with the oyster literature. This is a high salinity line, and this is a low salinity line that was actually developed from the New Orleans area. And we expected them to have high survival. And this is in part based on growers' observations, and also we had very early on had sampled oysters following a mortality event and found that both Sycamia and Virginica had low copy number. So what did we find? On the left here, we have the Australian variant. On the right, is the French variant. And we have, again, this is a 6-day trial, survivorship. What we found was that some of the Gigas lines, so line 1, which I actually thought would have high survivorship, and line 2 really had low survivorship through the 6-day experiment. But line 3 was relatively high survivorship against Australia and relatively low versus France. As well as the Sycamia, we've been calling them kind of the flip-flop lines. And to this really showed us the importance of screening with multiple variants of the virus to determine what the best species or stocks are. And also importantly, both the Virginica lines did quite well. We followed this with qPCR data. So every single animal in these experiments, qPCR was conducted on. And these are just, to orient you, are the Australian morts. And again, this is copies per milligram of tissue. In this case, since these were bigger animals, we took about 25 milligrams of tissue. And you're probably already noticing that all of the oysters that died to Australia have high copy number, maybe not as high as some of the tiny, tiny guys that we work with in their mortalities, including the couple that died in Crassostrea virginica. If we compare this to the survivors, both Aussie morts and survivors, and also French mort and survivors, a clear pattern is emerging that there is very low copy number in the survivors. In fact, there's 1,000 times more OSHV1 DNA in the mortalities based on statistical analysis. But this also made us question whether these animals are resistant or if they're tolerant. So these survivors might be more tolerant to infection. And so in future trials, we'd like to do some of these trials longer to see if they hold on to the virus. And something that I didn't mention earlier, which I often do, is that classically herpes viruses have active and latent infections. And we don't fully understand what might reactivate a herpes virus and cause them to start replicating and lead to mortalities. But this is one speculation that survivors carry low copy number before being reactivated. So we did an injection trial very similarly with 3 of the lines again, so CG1, 3, and that CV2. And we saw basically identical results where the CG1, the lines are on top of each other, was highly susceptible. CG3 was more susceptible to the French microvar. And again, very nicely, this Crassostrea virginica had no mortality. And we're actually doing an RNA-seq analysis of the 2 oyster species exposed to the French variant. So we're hoping soon to have some data to show you some preliminary stuff that the sequencing facility did. If we believe it, we could see some differences in expression between the 2 different oyster species. So the last technique, which I mentioned, is more of a natural exposure technique. In this case, we were working with little tiny oysters, which are much more able to become infected by a natural exposure. But again, the first thing we did, Tori is showing us, is we injected a bunch of kind of subadult large juveniles. Again, here, some of them gaping open. And then we allowed the virus to replicate for about 24 hours. And in this assay, we added little tiny oysters, again, these are like the fingernail size ones, to plates, and we added the water that came from being shed by the adults that also had antibiotics added to it, and we exposed them to each of the viruses. And this actually, we used 60 mils of infected seawater in each Petri plate. And what we saw, very interestingly, is, I think I skipped this on the last slide, is there was a difference in the amount of virus based on the variant. So this OSHV1 Tomales Bay, we consider to be a less virulent form, which is reflected in this lower level of mortalities across the 20 lines versus the Australian and the French. And in fact, statistically, that is the trend. And these were part of another study working with the molluscan broodstock program, which actually does oyster breeding on the West Coast. And they had previously done experiments in France with the same French variant, and we did some follow-up work. So they did 70 families. Thank goodness we only did 10 low and 10 high, because this is a lot of oysters to count. And then we zoomed in on 3 high and low performing family lines. So right here, these are the low performing family lines, and we can tell they relatively fit this pattern here, though maybe the spread isn't as much as in our colleagues' experiments. And we also looked at qPCR of individuals that were alive on day 4 and day 6. And interestingly, even though, and again, and here I'm just highlighting the Aussie and the French. Interestingly, even though, say, family 5, I happen to know family 5 was very clueless to what was going on and was a happy filtering oyster, and they got very infected. But statistically, all of this data was similar, except for day 4 was greater than day 6. And again, this is very high copy number, and this is copies per individual oyster. And right now, we've recently extracted several morts, which we expect to be very high in copy number, just to confirm this. So up until a year ago, I would have told you that OSHV1 was only detected in Tomales Bay and some of the neighboring bays. But in 2018, I was a colleague I work with who runs his own company, Ralph Elston at Aquatechnics, called me up and said, I had a detection of OSHV1. Can you type strain the virus? And they said, oh no, it's San Diego. There's a lot of shipping that goes in and out of San Diego, and with what happened with the other microvirus. And just to highlight one other thing, the West Coast industry goes from Washington clearly down to San Diego, though not as much culture in California, as well as Alaska and Hawaii. And there is a high health program that regulates the movement of animals, and there's a lot of testing been done, and that's how I can, Ralph has done a lot of this work pretty accurately, in my mind, say that these are our only 2 detection areas. And interestingly, this was a group of newly brought in oysters. And again, this was the first detection outside the Tomales Bay area in the US, likely since the early 1990s. And it was in a new aquaculture venture, establishing health history. So they had taken samples for 2 years and had no negatives. After this detection, just to add one more thing, is that they lost most of that group of oysters. So the ruling by the California Department of Fish and Wildlife was they depopulated and shut down the nursery. And the original seed source was tested, and a heat test of juveniles coming from the source bay was conducted. So it's been shown in France that they can bring in animals during a mortality period, increase the temperature, and invoke mass mortalities. So this is what was done. And they didn't see mortalities, and they didn't detect OSHV1. And there are ongoing sentinel outplants by the California Department of Fish and Wildlife to date, actually. So they're still trying to figure out if Pacific oyster seed can be grown in San Diego. And just, this is as they were depopulating this is the nursery system they were in. These big bins had lots of little juvenile oysters in them. So again, I was sent these samples, and I'm sorry there's a gel, but there's two things I'm going to point out about this gel. Number one, I'm mostly going to talk about this top part of this gel, which is for a specific genome region that they have used to be a diagnostic to differentiate variants. Whether this is the right genome region, that's a whole nother story. But just another thing is, I was so worried that someone was going to accuse me of contaminating this stuff. I was the only one that touched this, and I put everything we had in the lab. So I had recent DNA. I had the Australian variant that we were already working with, 3 San Diego samples, the French microvar, and Tomales Bay. And whether or not you're familiar with gels, you can see they all look really similar. And I also did another genome region that's recommended by the OIE, but I won't talk about that today. So again, highlighting this top section of the gel. And I had to go old school, so I had to go back to Sanger sequencing, and I was able just to use PCR products. And my 3 San Diego samples were all identical, beautiful sequencing data. And I went through, and again, I sequenced this French one, this Australian one, and here's also my 2003 one. And what I've highlighted here, these are all considered microvariants, and they are over 99% similar to this San Diego sample. So I've characterized this as a potential microvariant versus all of these other earlier samples that were classified as similar to OSHV1 reference strain. So this has clearly created an interesting situation. In fact, I was able to freeze in the -80 some of the animals that came from depopulating the Flupsy. And we did a pilot transmission study, and I just had a little bit of virus. I thought this is never going to work. But what we found out actually, so this graph shows some different oyster groups. Are either, the solid is either injected, the striped is a bath. This one, the bath oysters didn't get infected, and these two again are the same. And they were either injected with 25,000 copies, which earlier I described 1 million copies, or they were exposed by bath with a total of 260,000 total copies. And what we found were that, in fact, the virus appears to be quite infectious. So again, this is copy number per milligram of tissue, and these are the number of animals that died in the trial. Again, this was a pilot. And we used a couple of different groups. There was speculation that it was because the Flupsy planted triploids, and there was a couple of different sizes. And it's actually, these ones were really difficult to inject or to infect at all, and there is a size class that's much more difficult to infect. So what this told us is this is an infectious virus. And in fact, I mentioned that I had been in Tucson. So I was just in Tucson, and I don't have all of the results, so I'm not showing you yet more oysters, but we did 28 West Coast families, and we did 3 different viruses. We did the Australian, the French, and we did this one. And we, again, showed that we were able to transmit this, and this was through that more natural exposure route. So what are the future effects of this virus? I think one of our biggest questions still today is this question on the global strain variability and what does the genotype to phenotype mean? And I will be honest, it's not easy to work with these viruses, and we're very lucky to have something that's a tractable system that we're able to do these experiments with. It's also particularly, there's been a lot of rapid emergence of mortalities in some locations, which is troubling. There are also environmental factors. I mentioned temperature. Temperature is often related, though honestly we're the only people that we know of in the world that have multiple variants, so we might be able to go back and do some comparisons across temperatures. Salinity appears to be a factor as well, so I think some of the lower salinity estuarine waters of the East Coast, probably oysters wouldn't become infected based on some work in Pacific oysters. I've already mentioned several management strategies. I show you this because a reporter called me for a follow-up and I said, oh no, I can't tell you that OSHV1 is the latest symptom of global warming. We don't have data that supports this. And oddly enough, I get this sent to me fairly often. And so I put this here. I don't want you to think I think that, because there are so many other human cost factors going on here. Arguably, the movement of animals, the movement of water, probably through ballast water. So it's just one of these things that we really have to be careful about how we think about these issues. I mean, clearly there are lots of other things happening here. So what are the next steps needed? So we know that non-Pacific oyster species are less susceptible, but we need to look at more life stages and lines, which we were just doing, not only as looking to see if they're able to be infected, but also assessing them as potential vectors. We do need to additionally do more characterization of OSHV1 from San Diego. I think we'll be fully sequencing the genome fairly soon. Also to develop a high throughput assay, so probably a multiplex qPCR to differentiate variants. And lastly, I already described this gene expression analysis of the host. It'd be really, really interesting to see if we can find some big difference in the response. I'd also, big picture goal is to be able to pair our field and laboratory trials with quantitative genetics to really identify markers of resistance and susceptibility. And finally, really, the goal is to reduce potential for introduction. So the next time you buy oysters, if they're not from here, please don't put them in the water. That is can be a route of introduction of diseases. So think about, you guys are very smart, knowing scientists, so think about where you might put animals. Just to acknowledge, there's been a lot of different funding sources over the years, primarily Sea Grant as well as USDA. We work a lot with the wonderful folks at Hog Island Oyster Company, as well as lots of other members of the shellfish industry. And this is a particularly fun picture from Hog Island Oyster Company if you ever get a chance to go there. And thank you.